Once again, I welcome all the participants, and I'm really proud and privileged to invite and welcome Professor Sudipto Saha, the scientist working in Division of Bioinformatics, Bose Institute, Kolkata, a very famous, very reputed institute in this country. And Dr. Sudipto Saha is working in systems and synthetic biology. And to my surprise and wonder, he has sent a biodata. I asked one page biodata. Sir is so like metaphase chromosome. His contents his biodata into only 4 lines. So I could not able to understand much about this thing. But the biodata that he provided us really reveals information like he completed his PhD in immunoinformatics. Really, very few people are really working on immunoinformatics. In the year 2007, he got his degree from Jawaharlal Nehru University, and he worked in a CSIR MTech Chandigarh program, and he carried out or he underwent 2 postdoctoral trainings, one in Indiana University School of Informatics, Indianapolis, US, and from 2007 to 8, and then other postdoc in Case Western Reserve University School of Medicine, Cleveland, Ohio State, US, in the year 2008 to 12, and subsequently, Dr. Saha has joined Bose Institute as DBT Ramalingam Swamy Fellow in the year 2012, and now working as assistant professor. And with this a small brief introduction, I invite Professor Saha to take the session, for the benefit of young minds of this country. Dr. Saha, please.
Yeah, first of all, I'd like to thank Professor K. Balakrishnan for the kind invitation to give me a talk in this forum. And I welcome you again to the 4th day and 11th session. As I heard that you have already spent 4 days, I think you have already heard wonderful talks from distinguished speakers. So today I'm going to speak about systems and synthetic biology. So Professor Balakrishnan asked me to give a talk on systems and synthetic biology. So I thought that we do some work on systems biology, and we just initiated some work on synthetic biology. So today, whatever I'll be talking about, it mostly about the intercellular events within a cell and how things are happening within the cell. So when we talk about systems modeling or systems biology in India or across India, we talk about the protein ligand level, we talk about the genome-wide transcriptional or protein level, we talk about the pathway or the network level, and we also talk about the cell-cell interaction and the population level. So if you see this triangle, and if you see its triangle represents a pyramid, so most of the work at the base, it deals with the protein and the ligand level interactions. So majority people work in this particular area, and there are 2 ways you can study. One is the experimental method, and other is a computational method. So experimental methods are you use X-ray crystallography, you use spread, and as we all know that computational methods, we use docking and molecular dynamics to study the protein and the ligand level. So if you go a little bit higher up of the pyramid, we talk about the genome-wide transcriptional or the protein level. So here the experimental methods are the microarray, NGS system, proteomics, siRNA analysis, and metabolomics. And the computational methods used to study the genome-wide transcriptional or protein level are the clustering-based method, the network inference-based method. And if we go 1 tier up, there is a pathway at the network level. So here the methods generally we use are the metabolomics like mass spectrometry or the NMR spectrometry, microarray analysis, proteomics, also the siRNA analysis. And computational methods we use graph theory. So in today's talk, I'll little bit talk about the graph theory analysis, and we use an integrated based approach to study the topological of the pathway, and also we use quantitative based method. When I say quantitative, we are not interested to know only what proteins are involved. We are also interested to know what is the concentration of that protein or the concentration of the mRNA. So we use the term called expression or abundance. So if you go one level higher up, then we talk about the cell-cell interactions. For example, in immunology, we see that many cells like T cells, B cells, they interact. And so for that type of study, we use the dynamics or the microscopic techniques, fluorescence activated cell sorting. In short, it's called FACS. And computational methods are the Boolean-based method, the agent-based method, and we also use differential equation-based method. So at the highest level is a population level. So for example, we do study epidemiological studies like host population genetics, and the computational methods are the agent-based modeling, the differential equations, and the epidemiological models. In today's talk, I'll be concentrated on this red colored circle, mainly concentrated on the pathway and the network level, and the genome-wide transcriptional or the protein level. So if you talk about the intercellular events which happen within a cell, so there are at least three things can happen. One is the pathway. So in my subsequent slides, I'll talk about what do we mean by pathway, and then interactions, and then entity. So within an entity, there are genes, there are proteins, there are DNA, there are RNA, there are complex, and there are small molecules. The physical entity, suppose you have to understand one thing, that when you talk about gene, for example, MYC is a gene. So MYC within a cell can be present in a mRNA level, present in the DNA level, present in the protein level, and within a protein level, MYC can be present in a phosphorylation state. Suppose one of the side is phosphorylated, so in a MYC there are two phosphorylation events are there. So there are two sites where the threonine and the serine get phosphorylated. So suppose in a protein level also, the MYC can be present in an unphosphorylated state, and in one site phosphorylated state and two sites phosphorylated states. So you have to understand within a cell, MYC can present in a mRNA level, DNA level, and in protein level. And within a protein level, there may be some phosphorylation events can take place, and there are different species can take place within a cell. So coming to the interactions. So interaction is a very broad term. So generally we talk about the genetic interaction, we talk about the molecular interactions. So today's talk I'll be concentrated on the molecular interactions. There's a conversion, as we know that the enzyme substrate complex, then there's a control mechanism, there's a catalysis mechanism, and there's a template reactions. So, and what we just I want to give a bigger picture to you, that within a cell, suppose we talk about T cell, and suppose within a T cell, suppose there are different population of T cells, for example, CD4, CD8, Treg cells. So within a T cell population, you see in the intercellular events, there are so many genes, so many proteins, so many metabolites are there. So if we can we understand what are the events taking place within a cell? So the question comes is that can we model the intercellular events? So there are two approaches. First is the top-down approach, which people generally use it. So where you take all the data from the physiological data, the tissue level data, and then you try to model it. But due to the advent of next generation sequencing and the proteomics like mass spectrometry, people use bottom-up approach. People generate the high throughput way, all the genomics data, proteomics data, the metabolized data, and then they try to model it. And it's coming from the left hand to right hand end. The thing is that why we try to model pathway instead of a molecule, because a single molecule have a very short life. For example, MYC has a degradation of maybe around 30 minutes, or it has a half-life of 30 minutes. And if we try to study MYC, because MYC can be degraded at any time within a cell, and so it's better to study what are the proteins where MYC interact, and what are the proteins MYC interact both at the DNA level, at the protein level, and can we study that particular interactions. So people started modeling from the bottom-up approach instead of top-down approach, and try to model the pathways. So coming to the genomics. So now I'll briefly touch about three high-throughput omics, mainly the genomics, proteomics, and the metabolomics. And then I'll try to give a bigger picture how we integrate the genomics, proteomics, and the metabolomics data to get an interactome. So when you mean interactome, interactome means all the species. Here, the species are the protein, genes, the mRNAs, and how they interact upon each other. So coming to the first high throughput technology, which is the genomics, people generally use it very often. And the whole idea is to get the global gene expression profile. And here, the two type of transcripts we measure, one is the mRNA and one is the miRNA. So that there is an interaction between the mRNA and miRNA. The interaction is a negative association. So if mRNA binds to the particular mRNA, they control it and they try to inhibit the expression of the mRNA. So to study this, people use microarray, next-gen sequencing, and within a next-gen sequencing, there is a first generation, second generation, and the third generation. Nowadays, we come up with the third generation next-gen sequencing. So I'll briefly talk about little bit about the third gen next-gen sequencing in my subsequent slide. The whole idea of genomics is that you get your reads, small reads, and then you try to align it with a reference genome, and then you try to quantify it. So when you mean quantify, we try to mean the expression, the abundance, how much they are abundant. So the whole idea is to first align it. When you align it, you'll come to know the what type of gene it is, and then we try to make a quantification that gives us how much abundant a particular gene is. The good point of genomics study is that the coverage of genomics study is very high. You can at least get 100% coverage. Suppose if you want to study the next gen sequencing and using the second generation or the third generation, you will try to get a global picture. So when I mean global picture, suppose in humans there are approximately 30,000 proteins. So you will get approximately the whole overall idea of 30,000 proteins as well as the transcripts and at what level they are expressed. Unlike in proteomics, you don't get the global type. Although we try to study proteomics in a global level, but the methods available to study the proteomics doesn't allow to study all the 30,000 genes present in the human. So it's always good to study with a genomics study. So what we can do with the gene expression data? Suppose when we do genomic studies, the whole idea is that whether some genes are induced or repressed in a developmental phase or an environmental change. Suppose we give some stress or we do some drug treatment to the experimental model, for example mice, and try to see in a blood whether some genes are going up or some genes are going down in giving a treatment or a particular drug in an animal model. Also, we set up genes that are rise and fall at the same condition. Suppose if you give some condition, suppose you give some environmental stress and see whether two genes are going up or going down at the same time, or they are going in a different directions. That gives us an impression that whether those two set of genes have a related function or not. For that, we use the time scale gene expression data. Third is that, which is very important, is that some common regulatory motifs are there, which we call as the transcriptional binding sites, and there are some protein-protein interaction taking place. So when we see the set of genes having or they are co-expressed at the same time, they are most likely they have a common transcriptional factor. So in that way, we can try to find the regulatory motifs, and that help us to study that whether a transcriptional factors bind to a regular motif and whether they are co-expressed at the same time or not. Finally, we all know that the use of gene expression is that in biomarkers, and biomarkers are if you want to study an indicator gene or indicator protein in a particular disease state, for example cancer or for example asthma, we use an indicator protein and we study the gene expression, and for that way we try to find out whether a particular gene can be used as a biomarker or not. So for the global gene expression experiment, you have to design your experiment and then you have to do some statistical processing and analysis. And suppose you have three conditions and using the three conditions, if you want to study whether there are a set of genes are there which are expressed at a different conditions. For example, I give an example of suppose CD4, CD8, or the Treg cells. You have a three population of the T cells and try to see the global gene expression and try to see whether there are some set of genes are there or not which are expressed in all the three populations, and then you see whether a set of genes are expressed only in one condition but not in the other conditions. So in that way, genomics gives us a complete picture about the whole genes present in a genome level. And at the end of the day, we do clustering based method. It's a machine learning tool, and it's a way we cluster the genes, and based on the cluster, we assign the functions, that whether a particular set of genes with a cluster together, whether they have similar functions or not. So in that way, we cluster it and get the functions of the particular set of genes. So coming to the proteomics. So proteomics, as I was talking to, that proteomics is a study of large scale study of proteins, but unlike genomics, we cannot study all the possible proteins available in the human gene using the techniques which are available nowadays. For example, there are 30,000 genes are available in a human, for example. So if you do proteomic study at a time, you can at most study 10,000 proteins at a time. You cannot study all the 30,000 proteins at a time. Unlike in the genomic studies where you can study all the 30,000 possible genes and the expression using the next-gen sequencing techniques. So the term proteomics was first coined in 1997, and it was analogy with the genomics, and where in genomics we study the genome. So similarly, proteomics study the whole proteome. And we use the tandem mass spectrometry, and we use a database search engine. We try to identify the proteins from the complex mixture. And again, in proteomics also, we try to differentially express whether a set of proteins are differentially expressed or not based on the quantitative proteomics. And then we also study the phosphoproteomics. This is a very important thing which we can study using proteomics, where we cannot study in the genomic study. In phosphoproteomics, many proteins, as I was giving an example of MYC in the first or second slides, that a MYC can present in a one-level phosphorylation events and two-level phosphorylation events. So the protein, when they are phosphorylated, they get some cascading events takes place, and the interaction level changes whenever they are phosphorylated or they are unphosphorylated. And finally, we study the interaction proteomics, which is very important. So as we know that we study the protein-protein interactions, and most of the protein-protein interaction nowadays are studied using the interaction proteomics, where people use affinity purification followed by the mass spectrometry-based methods. So the question comes here that why we study proteomics, why we cannot study with single gene at a time. So suppose if you study single gene at a time, then it will take a huge amount of time. A particular department or a particular lab can study a single gene, and then they spend 10 years, and then they get information about a 2 or 3 genes at a time. But if we use the proteomics-based approach, suppose the Department of Genomics and Proteomics, you can study a bunch of proteins at a time. For example, if you want to study the cancer, and you want to study which are the proteins which are involved in a particular cancer state as compared to the normal state. So you use a proteomics-based approach, and you study a bunch of proteins, and then you pick up one by one and try to find out whether they have their role, their informations, and whether they are indeed involved in a particular cancer. However, there are some concerns when you study proteomics, that if you use a net-based method, suppose, for example, I'm just giving a small example. Suppose you use a net, and the whole idea is to catch a fish. But sometimes instead of catching a fish, the small snake can come within your net. The whole idea is that you have to remove the small snake from your net. So similarly, when you use a proteomics-based approach, there may be a possibility some false positive proteins may come, and they may or may not be involved in a particular cancer state. I'm giving an example of cancer. So the whole idea is that when you use proteomics or genomics, you may get a bunch of proteins, and then you have to fish out that whether all the proteins are involved in a cancer or not. So in that way, use some statistical based methods and remove the false positives from your set of proteins and try to find the true proteins or the true interactors in your studies. So for that, we use a statistical based method and bioinformatics tools to remove all the false positives from your study. So when we use genomics or proteomics studies, so statistics and bioinformatics play a very important role. So coming to the proteomics, as we know that proteomics represents the effort of it identifies the protein, it quantifies the protein, you can get the structure, you can get the biochemical and the cellular functions of all the proteins in organism, organ or organelle. The important thing about proteomics is that as we know that the proteins are the most downstream molecule in a cell, and their functions are very important. So they vary in space, the time, and the physiological state. So unlike in a gene, it's more or less constant in a cell at a particular time, but in a protein, they, as I was talking about, they get phosphorylated. There may be some phosphorylation events takes place, and then they get degraded using the ubiquitination based methods. So they're very uh, dynamic in nature in proteins as compared to the gene. So coming back to the proteomics-based approach, so we use 1D or the 2D gel electrophoresis-based method. We use protein identification, and we also study the post-translational modification-based method, and we use mass spec to identify the protein, to quantify the protein, and also try to find out the post-translational modifications. So what we do generally, we use, first of all, we use the separate the proteins based on the charge, the isoelectric point, and then we use the STS page. So I think it's a pretty straightforward based method. And first of all, we start the protein samples, put in isoelectric focusing, and then perpendicularly put it in STS page and try to find out the proteins. And then we do 2D based proteomics. And this is how we get the proteins overall across the thing. And if you see that 3 to 10 is a pH gradient we use, and these are the masses of the protein, you can see it. The other method is a peptide-based fingerprinting based method, where we run the protein in a gel. It can be 1D gel, it can be 2D gel, as I was talking about. You excise the protein and then use trypsin. So there are many other enzymes, but we generally use trypsin. And trypsin is an enzyme which has a specific chopping site at the lysine and arginine, and it chops the protein into peptides. So what we do in case of proteomics based method, we get the protein or the complex mixture, we chop it peptides, chop the protein using trypsin or pepsin and put it in a peptide level form, and then we use MALDI, and using MALDI we get the peptides, and then we put it in a computer, and in a computer we have the database where you have in silico cards of lysine and arginine, and try to match the experimental chop peptides with the in silico chop peptides, and then we come up with the protein level. The other method is the most sophisticated based method, which is the state of the art nowadays, where you use MSMS based method. First of all, we get the peptide, and then again we bombard the peptide and put it in an amino acid level and get the sequence of the amino acids, and then put again search in a database and try to identify the proteins. So next I'll little bit get up a little bit about the proteomics study and how people use the proteomics, phosphoproteomics to study the intracellular events. So I just took a paper from PNS, and here they studied the CD28 receptor, which is a co-stimulatory receptor in a T cell, and they tried to study what are the intracellular events taking place within a T cell. So T cell contains the T cell receptor. Along with that, they have the co-stimulatory molecule, which is the CD28. And the TCR and the CD28 co-stimulatory molecules, they work together, and whenever they get a signal in the signal coming from the CD28. There are cytoplasmic tail is there in the CD28, and some cascading events takes place in the cytoplasmic domain of CD28, and then there are a lot of interaction and intercellular events taking place within the T cell. So in this paper, they talked about what are the intercellular events taking place whenever there is a perturbation in the CD28 co-stimulatory receptor present on the T cell. And they try to identify the intercellular signaling events taking place within the T cell. The importance of CD28 as well as the TCR is that they regulate a lot of interleukins, generally interleukin 6, interleukin 2, and they play a very major role in the disease conditions. So if you see the CD28, so it has a long cytoplasmic tail, and within the cytoplasmic tail there are three motifs are there. First motif, which is YMNMM, second motif is a PYAPP, and third motif is a AYRS. And in all the three motifs there are some phosphorylation sites where the phosphorylation event takes place. So in this paper, what the authors have studied, they try to see what are the interaction taking place in the cytoplasmic domain in the three motifs. So there are three motifs are there. They individually try to see what is happening in the first motif, what is happening in the second motif, and what is happening in the third motif. So if you see this particular membrane, and this is a cytoplasmic domain, and whenever there is an event taking place, there are so many interactions taking place. And this is what they study the pathway analysis, because there are so many events are taking place. See, suppose in this particular PY3K, there are a lot of events taking place, and then again in this particular motif, so there are events taking place by a single motif, there are events taking place by the two motifs. So here, this particular GRB2 here, the two motifs they interact, and this particular event that takes place. So, and the third motifs, we can see that not many interactions taking place. So if you want to see that, get an idea that whether a single motif is important or whether the combination of two motifs are important to see the cascading events. So they did some phosphoproteomics study and tried to generate the data, and they performed the study. And what they found is that, see, a lot of pathways are involved. The first pathway is the Wnt signaling pathway. As we know that Wnt signaling is a very important pathway in the developmental biology, and we'll talk about a little bit about the Wnt signaling pathway in my subsequent slide. So this particular event induces so many pathways. You can see there are a lot of pathways are there which are induced by only a single event. So what they did in this particular paper, so instead of looking for the two cytoplasmic motifs, they did some synthetically they designed the motifs, and they used some phosphorylation events within the motifs, and instead of studying the three motifs at a time, they studied the two motifs, and they chemically induce the phosphorylation events at different positions, and they synthetically design the cytoplasmic domain of the CD28 and try to study the SILAC way, which is a study where people use the isotopes. In this case, they use the lysine and arginine, and they use the lysine and arginine as an isotope and use medium and heavy of lysine and arginine, and then they try to find out that which particular site the events are phosphorylated within the motif. So what they found in this particular B panel, if you see the B panel here, so they try to find out what events takes place when the two motifs are phosphorylated, whether there are any events where two motifs get together and they perform together. So what they did, they used a table and they found out the list of proteins in this particular section. They found out that whenever the two motifs are phosphorylated, then these particular proteins, they get expressed and they interact. In panel C, if you see there is a 1, 2, and 3. This is the first motif. So these motifs, they interact with this number of proteins, This second motif, they interact with this number of proteins. And as I was talking about my previous slide, there was a third motif. And third motif, there are a very small set of proteins which interact with the third motifs. So in this way, they systematically study the role of interactors of all the motifs in the cytoplasmic domain of CD28 receptor of T cells. Finally, they try to study which are the pathways which get involved in CD28 whenever there is a event taking place. So first of all, they found that the T cell receptor signaling pathway, they go up, the natural killer cell events take place, and in this way they found out there are many pathways which are involved in the T cell events, they get activated. So finally, they study the interactome of CD28. So what they did, they studied the phosphorylation dependent CD28 interactome. So if you see the CD28 here, and if you see the outer circle, this is the outer circle. The outer circle are the set of proteins which directly interact with CD28. So these proteins which are coming in the outer membrane of the circle represented here, they directly interact with CD28. And the inner circle, these are the set of proteins which interact with the outer circle proteins. So these inner circle proteins, they do not interact with CD28, but they interact with the interactors of CD28 proteins. For example, if you take this particular complex, they interact with the PIK3R3. So these proteins, they do not interact with CD28, but they interact with PIK3R protein, and PIK3R protein, they interact with CD28. So in this way, they get to study the interactome of not only the direct interactors of CD28, but also the indirect interactors, which is represented here of the CD28. So their next goal is to study that whenever these set of proteins, when they interact, whether they have any common function or not. Because at the end of the day, we are interested with the function of the particular set of network or pathway. They came up with very interesting results, mainly the actin cytoskeleton organization based events. So whenever there is some signaling taking place in the CD28 co-stimulatory receptor of T cells, that there is some actin cytoskeleton organization taking place, and there is a positive regulation of the biosynthetic process. There's apoptosis taking place, there's a protein complex assembly, regulation of the GTPase activity, and there are some intercellular cascading events. So one single event which is happening in the surface of the T cell, which is a co-stimulatory receptor, so many events are taking place in the cytoplasmic domain, and we can see that the intercellular events are taking place. So here they try to study all the intercellular events taking place within the T cells whenever there is some signaling coming up from the CD28 of the CD28 of the T cells. Coming back to the metabolomics, as we know that there are some low molecular weight compounds called the metabolites like glucose and fatty acids. They are equally important besides genes and proteins. So here we talk about the term called metabolome. So here we refer to the complete set of small molecule metabolites, and they are very dynamic like proteins. And metabolites, we all know, we studied from at least from the 8 or 9 class 8 9 book that there is a Krebs cycle, there's a glycolysis pathway, and all the metabolites, they play an important role in the Krebs cycle, like the carbohydrates, fatty acids, amino acids, and most of the amino acids and the secondary metabolites, they synthesize using the particular cycle within our cell, and they get synthesized. And the techniques are to study the metabolites are the gas chromatography, HPLC, NMR, and also you can use the mass spectrometry based method, as I was discussed in the proteomics based study. So the question comes is that if you want to study, or if you have a biological question in mind, then how to start using systems and synthetic biology. First thing is that you have to see whether similar type of work has been done, or people have already done, or people have already addressed or not. Then you can go to the database and try to fetch the data. For example, in case of next-gen sequencing, or microarray, there is a database called the Gene Expression Omnibus. You can put your biological questions, what you have in your hand, you can put it and see whether similar type of study has been performed or not, and try to dig the data and try to analyze the data. You can always go to the PubMed. You can search the biological question and see whether similar type of work has been done or not. For the protein function and annotation, we use SwissProt, and then we use Ensemble. But the question comes here that there are some advantages and there are some disadvantages. The advantage is that it's freely available. You don't have to pay from your pocket. You can get already some data are available. There are some raw data, there are some normalized data, and there are some already developing tools and databases are available. But the disadvantage of using the publicly available data is that suppose you have a biological question in hand, the study which has been already done, it may or may not directly or 100% matches with the aim and objectives of your study. So you may find some similar study, but it may not be the 100% the same biological question you want to address. For example, people do meta-analysis and people do data integration studies, but it is very difficult to get data from same samples across different experiments. Suppose you have a biological question in hand, and you try to study 5 or 6 studies which has been already been done and available in the databases or in the PubMed, and try to get the data, and maybe your biological question may not be directly matched with what type of data already available. So for that, what you have to do, you have to generate your own data. And I would like to talk about the human protein interaction database, HPRD, and it deals with the protein-protein interactions, it deals with the PTMs, it deals with the protein expressions, subcellular locations. So it's a very important source of studying the protein-protein interactions, and all the data are experimentally designed using APMS-based study, which is the affinity purification, followed by mass spectrometry or is2 hybrid-based method. There is another database which is called String, where it talks about known as well as the predicted ones. So many times you try to find out a protein where the experimental protein-protein interaction data is not available, but you are interested to know what are the interactors of that particular single protein. So for that, you can use String, and you can use the co-expression. I was talking about the co-expression. Based on the co-expression, you can study the protein-protein interactions. So in this way, the String allows you to study not only the experimentally determined protein-protein interaction, but also it helps the experimental researchers to try to find the predicted protein-protein interactions. So in our lab, or in most of the systems biology lab, they try to study if they have any biological question in mind, they try to generate their own data, because as I was saying, that the biological question what you want to ask, it may not be exactly reflected in the data which is available in the public domain. So for that, you have to generate your own data in your lab, and in that case, the mass spec based method allows you to study the proteomics set data. I discussed about the proteomics, how you do, like use the protein, you use gel first of all, get your protein, chop it. You may use MALDI, which is a very straightforward, but the state of the art is the tandem MSMS. First you get your peptide mass fingerprinting, then you bombard each peptide to get the sequence of the peptide, and then you search in a database, you get your protein. So coming back to the protein-protein interaction, which is very important because most of the cellular events takes place at the protein-protein interaction level. So you have your bait. So here bait is your protein of interest. So you want to study the interactors of your protein. So here we term as a bait protein. So bait protein, what we do generally, we put a tag. So it can be epitopes like DYKKK, it can be 3 tag, or it can be GFP proteins. Then you can put a tag to it, and then you express in a cell line. Once you express in a cell line, you have your tag, and then you use an antibody which have an affinity with your tag, the epitope, and then you pull down all the proteins which interact with your bait protein, put it in a mass spec, try to identify the peptides, and then quantify the protein, and then you got your protein, and then you try to assemble it in a that which protein interacts with bait or which protein interacts with within themselves. So there are two types of interaction can take place. One is your bait protein directly interacting with the prey proteins, and there may be a prey proteins. The prey proteins are the interactors of the bait proteins. So prey proteins may directly interact, they may not interact directly with the bait proteins. So there are two types of interaction can take place, and you want to study the complex. So within this complex, again, there is a term called the false positive is there. So when you perform this type of study, you may get some false positive proteins are there within your complex. You use some statistical based method, try to remove the proteins which are not directly interacting with your bait, and then you get your complex. So the third thing, as I was talking about the genomics based study, and with the advent of the third generation sequencing, which is a mini ion, which you can procure in your own lab, it costs only 4 lakh rupees, and then you can study at least the microbial studies. You can do it very easily in your lab, and you can generate the transcriptomes. You can use the 16 ribosomal RNA, you can use the SNPs, and try to generate your own data in your lab. There are some other next-gen sequencing based method, but generally like Illumina or the 454 based platform, which are the second generation based technique. So this requires a lot of money. Mainly the machine is a weak machine, and there are a lot of you have to maintain that machine, and you need a technical person to run that particular machine. So but however, if you use Minion, which is a very small, like you can put it in your own hand size machine, there is no maintenance of AMC. You can generate your own data, and but the issue is that in case of Minion, there are some false positive error rate is little bit higher as compared to the Illumina, but that you can compensate depending upon the biological question you have in your hand. So we in our lab we use mass spec, we use Minion based method, and use protein based method, protein interaction based method, which is like a affinity purification, like putting an antibody which is targeting the epitope, getting the complex, put it in a mass spec, get the interactors, and then model the interactors to remove the false positive and get your true interactors which are interacting with your bait protein, which is a protein of interest. So coming to the protein interaction network, so there are, I hope you all come up with the term called network and pathway, but just for simplicity's sake, I just want to define it. So the networks is a collection of two or more proteins, here in this case proteins, that associate to form a functional unit. So it has to interact and they have a common function. So if they interact and if they don't have any function, it doesn't mean anything. So it may not be a network or complex. But when you talk about network or complex, it means that two or more than two proteins, they interact and they have a functional unit. So in this case, here the node, the green color, the size depends as node, which can be, for this simplicity's sake, I'm taking as a protein, and it interacts with another protein which is called edges. So in a graph theory, we define it as a node and edge, and in this way, it represents as a network. But when you talk about pathway is a bigger picture. So suppose I was talking about the Wnt signaling. So suppose Wnt is a glycoprotein which binds to the frisel receptor which is present in the membrane, and then that there can be two events can take place. One is in the presence of Wnt and one is the absence of Wnt. So let me talk first what happens in the presence of Wnt. Whenever Wnt binds to the frisel receptor, then the beta-catenin forms a complex. So this particular complex is represented here. You see the axin, is there, you see the APC, here is represented. So this is a complex formation and this is a network formation present in a graph theory. And you see the GSK3B and you see the GSK3B. So they form a complex like this, and then beta-catenin when binds to this set of proteins and form a functional unit. So I was talking about a functional unit, and they form a function, and then beta-catenin becomes stabilized, and they enter in the nucleus, and then they target the DNA, this particular complex, and then the cascading events of gene take place and a lot of gene get expressed. which plays an important role in the developmental biology or in the cellular events. But what happens in the absence of wind? Whenever wind is absent, this particular complex, the beta-catenin, there are some phosphorylation events takes place between the GSK3B and the APC protein, and the beta-catenin become degraded. And once beta-catenin get degraded, they cannot enter the cell, and then you don't see the cascading event taking place. So a wind which is coming from the extracellular matrix, when bind to the frisal receptor, the so many events taking place in the intercellular part. So one you can depict it here, which is in the presence of wind, and one is absence of wind. So the overall goal of the systems biology is to integrate all the single omics layer and to get the overall integrated omics. Suppose I was talking about the genome, I was talking about the transcriptome, I was talking about the proteome, I was talking about the metabolome. So for example, if you talk about the genome, there may be two events. First is some genes may be methylated and some genes may be unmethylated. So whenever there is a methylated genes, they do not play, or they are not active, and they don't play important role in the next events or the cascading events. So within the transcriptome, there are two transcripts, at least two transcripts. It can be more than two transcripts, because in mRNA there are miRNA, there are LNC RNA, there are piRNA. But for the simplicity's sake, I'm talking about two things in the transcriptome. One is the mRNA and one is the miRNA. So as I said in my previous slide, that mRNA and miRNA, they are the transcripts and they have a negative association. So in case of transcriptome, you can see there are mRNA, miRNA, and in case of proteome, there are two types of protein at least. One is the signaling proteins and one is the transcriptional factors. So transcriptional factors, as we can see, that represented in two different nodes. One is in a oval shape and one is in hexagon shape. One is in green, which is a transcriptional factor represented here, and these are the signaling proteins which interacts with the among themselves, or it can interact with the transcriptional factor as well. So these transcriptional factors, they can directly interact with the DNA, and they interact with the unmethylated part of the DNA, and then they induce the cascading events or they express certain genes. And again, in case of metabolome, you can see there are a lot of metabolomes are there, and they directly interact with the transcriptional factors. So if you integrate the single layer omics and get it in a integrated in one layer, you see there are all the features which is coming from the proteomics, genomics, and the metabolomics, and you see a lot of interaction taking place. So here there are two terms you can see. One is the solid lines. You can see there are solid lines. These solid lines are the interspecies interactions. Interspecies means the proteins which interact, the signaling proteins, they interact upon each other. And one is a dotted line. So dotted lines are the interspecies interactions. Suppose protein DNA and then protein metabolite. So you can see that there is a crosstalk between the multiomics layer, which cannot be determined by a single omics. So people in a lab, they want to study one omics like genomics or proteomics. But if you want to study the interactors or the interactome of a particular event, what is taking place, if you put an external influence, for example, I took an example of CD28 receptors, co-receptors in a T cells. So you can see a lot of events taking place, and there are a lot of interactions taking place. So suppose this integrated approach can be studied in the T cells, epithelial cells, macrophage cells, mast cells. And the question is that when you put an external influence, whether these interactome have a different set of interactions between the disease state or in a healthy state. For example, in a cancer, for example, suppose we take an example of Treg cells. So in a Treg cells, we know that the FOXP3 expression gets higher up and they goes to the close to the cancer cells. The question comes is that if you want to target an interaction, then you can use the small molecules or the monoclonal antibodies, which are called the biologics, and you can stop this particular interaction, and then you can stop the cascading events starting from this particular event. So the overall idea is that if you know the interactions, it allows you to use the next stage of identifying the therapeutic targets, because there are many transcriptional factors like MEK or FOXP3 which you cannot directly target it. You have to use an indirect based approach, like targeting the interactions rather than targeting the whole protein as such. To study the interactions, you have to understand the holistic picture of a cellular events taking place within a cell. So similarly, as I was talking about the T cells, so now the question comes that what type of immune cells you can be engineered. So as we know, in case of T cells, and in case of B cells, T cells, there are some receptors are available like CD28. But unlike in dendritic cells or natural killer cells, they are the part of the innate immunity, and then they don't have a specific receptors within the cells. But however, the natural killer cells and the dendritic cells and the macrophage cells, they can be tweaked by some using the interleukins. So there are some specific interleukins which acts like a outside the cell, and they can target the receptors which are present on the macrophages or the dendritic cells and not the natural killer cells, and you can target the receptors of the interleukins and the interleukins between themselves, and you can manipulate those things for your study. So coming back to the engineered T cells, people mostly studied the chimeric antigen receptors against cancer, and there are some tumor infiltrating lymphocytes, so you can express the receptors outside the cells, and you can use at least 10 kb of the genes within a immune cells, and you can express it and put it in a extracellular surface which can target the antigen present in the particular cancer tissues. For example, the most widely studied engineered T cells are the chimeric cells and the multiple receptor T cells. So here in this case of chimeric antigen receptor, which is represented as a curve, and the multiple receptors, they express a receptor which are very specific to particular antigen present on the cancer cells. So in this way, these particular engineered T cells, they can specifically recognize the cancer state and they can kill it. So coming back to the databases of multi-omic studies, I was talking about the multi-omic studies where you have different layers, a single layer, and then you combine the single layers to form the multi-omics layers. So suppose for the people who are interested on the cardiology, then there is a database called the CardioGene base, which is a specific database where a literature of multi-omics database of mainly the cardiovascular disease are available. CKD-DB, an integrative multi-omics expression database of the chronic kidney disease. If our people are interested in a kidney disease, then you can search the CKD-DB. Link, which is very popular one, a library of integrated network-based cellular signatures. Linked-omics is also very popular in the field of cancer. People use clinical proteomics tumor analysis consortium, and the TCGA people used linked-omics. MOPED, a freely accessible multi-omics expression database, and the TCGA, which is the database where all the gene expression, methylation, copy number are available across 30 different types of cancer. Now the question comes is that if you have your data, suppose you have your gene expression data, you have your DNA methylation data, or if you have your proteomics data, how to integrate them. So there are already softwares and tools are available which allows you to integrate the different omics layers. For example, the Bio Wardrobe, which allows to integrate the mRNA expression and the DNA methylation. Dingo is a very popular one. The mRNA expression, DNA copy number, methylation, microRNA expression. Immunonate, people who are interested in immunology, this particular software allows you to integrate the mRNA expression, protein-protein interaction, mRNA motif profile, and the transcriptional factors profile. There's a IMPLA, There is a J-active module. So in my subsequent slide, I'll take an example of J-active modules. We use J-active modules in our lab where we try to integrate the genetic and the protein-protein interaction network. And we get our mRNA expression, and then we compile with the protein-protein interaction data to get our network. So I'll take an example of the J-active modules in my subsequent slide. Lemon tree, which is the integrative multiomics modular network interface, Medscape2, metabolite and the gene expression data. People who are interested in study the metabolome and gene expression, this particular software allows you to integrate the metabolites and the gene expression data. Mixomics, mRNA expression, DNA methylation and protein, network analyst, gene expression data, protein-protein interaction data, omics integrator, pentomics, and the most important is the Zika virus. So suppose people are more interested in Zika virus, they have a software called Zika VR, which allows you to integrate the gene, protein, mRNA interactions. So depending upon your interest, you can use a particular software, and depending upon the data you generated in your own lab, or the data which you can use it publicly available, and you can choose a particular software and you can allows you to integrate the data and get a multiomics interactome information. So coming back to the J-active module, which we use very often in our lab. So we do the mRNA expression or the transcriptomics. In my beginning of my talk, I was talking about the genomic study. The good thing of genomic study is that it allows you to study all the possible genes and all the possible information you will get it. For example, human, there are 30,000 genes, you will get all the 30,000 gene information if you use the genomics data. So the genomic studies, suppose we have a control state and you have a disease state and we get the differentially co-expressed genes. Now we have a protein-protein interaction network. So these are the static network which represents the yellow color node, and each node represents other proteins. So in this case, you see there is no expression is there in the protein-protein interaction. So if you have a data where you don't know the expression of the protein, but you know they're interacting. So in that case, what you can do, you can merge the interaction network, which is a very static network, and merge with the differentially co-expressed genes and make it as a dynamic network. So when I mean dynamic means, suppose now this particular network, which was a static where you don't have any expression information, we have only the interaction information here. So but however, when you merge with the differentially expressed genes, we get a network where we have your interaction profile and also you have the expression profile. The beauty of using the merging the two different layers like gene expression and the network information, that you get a circle here which is represented as a green color here. You get a set of proteins which are interacting and which are having a similar type of information which is represented as a blue color. Suppose if blue color represents that they are expressing at high level. So in this case, in this network, so these set of genes or the proteins which are interacting and they are expressing at a very higher amount. So in this particular network, we represent this particular set of proteins as an active network. Active means they are differentially expressed, they have a similar type of expression, and they're also interacting upon each other. So we can target these particular active networks for your further studies. So I'll briefly talk about the graph theory, which we generally use to study the protein-protein interactions. This is the same picture I showed you in my previous slide, which is the APC complex and forming with the beta-catenin. And you see there is edges. I talked about edges. I talked about nodes. So there are 5 different terms people use. People use a half protein. So half proteins are the proteins. Suppose, for example, if you take an example, APC. APC binds with Wnt1, Axin1, CTNNB1, BAB1, this one. So 1, 2, 3, 4, 5, 6. So this APC has a degree of 6. So degree represents the number of neighboring interactors. For example, if you want to study the degree of BUB1, so BUB1 has a degree of 3. Why? 1, 2, 3. So you understand the term called degree, you understand the term called age, you understand the term that called node. So what are halves? Hubs are proteins which have a degree of greater than 5. So Bob has a degree of 3. So Bob is not a hub. So APC has a degree of 1, 2, 3, 4, 5, 6. So APC has a degree of 6. So APC is a hub. So we can define hub as a node. Here node represent protein where we have more than 5 degree. So then you can define as a node, and then finally when you talk about the biological network, we use a term called scale free. So what does it mean? It means that if you use a degree which is in the x-axis and if you use a node in the y-axis, then most of the proteins have a degree of 1 or 2. So they have a degree of 1 or 2 and they have degree of 1, 2 and 3. But when you go up with a degree of 5 or degree of 7 or degree of 10, you will find very less number of nodes which have a degrees of greater than 5. So it means that whenever the degree is higher, we have a little less number of nodes. So this we represent as a degree distribution follows a power law. So when we talk about the biological networks, to find out whether it's a biological network or whether it's a random network, first you have to perform a map which shows that whether it's a scale free or not. For that you have to understand the degree, you have to calculate the degree, and you have to put it in a distribution and see whether it's scale free or not. For example, many times we get a network which is a synthetic network. So when you use a synthetic network, so in that case you won't see a scale free network. So it will be something like this, it will be something like this. So it won't be a exponential power law, it won't follow this particular part. So whenever we talk about biological networks, first of all you have to see whether it follows a scale free or not. So next very important term is the clustering coefficient. I was talking about the bait protein in case of APMS, which is represented as a blue color node here, and it interacts with the 3 white nodes, which is represented as a prey proteins. So if you see, there are some solid lines and there are some gray color lines. The solid lines means these prey proteins, they interact upon each other. So this is a bait, which is a protein of interest, and you want to see whether they interact or not, and then you find out there are 3 prey proteins which interacts directly with your bait, and these prey proteins, they also interact upon each other. So there are 3 pairs and there are 3 interactions. So 3 out of 3 is 1. So clustering coefficient is 1. Suppose the next scenario is that this bait protein, you have the 3 interactors, and out of 3 interactors, there are these 2 prey proteins, they interact, but there is no interaction among themselves. So in this case, there are 3 nodes, the prey proteins, and there is only 1 interaction. So it's 1 by 3. The clustering coefficient is 1 by 3. And in the third scenario, you see the bait protein, you see the 3 interactors and they don't interact upon each other. Clustering coefficient is 0. So now you can ask me why we measure clustering coefficient. The clustering coefficient, which is represented as C, is very important. It allows to identify whether the prey proteins, they interact upon themselves or not. So whether it's a dense network or whether it's a free network. So suppose in this case, if your clustering coefficient 0 means you identified prey proteins which bind to the bait protein, but they don't interact upon each other. But however, in this particular case, we have all the prey proteins which are the interactors of the bait protein, and they interact upon each other. So next particular term, we use shortest path. So suppose, for example, there are 6 ways, 1, 2, 3, 4, 5, 6 bait proteins, and they interact upon each other, as shown here. Suppose you want to go from 1 to 6. You can go 1, 5, 4, 6. This is one way. Or you can go 1, 5, 2, 3, 4, 6. This is another way. And the third way is that 1, 2, 3, 4, 6 way. The whole idea is, can we identify the shortest path from one node to another node? So at the end of my talk, I'll just use the cytoscape tutorial just to show that once you get your all interactions and how you represent it in a cytoscape tool. So it's a very, if you can download the cytoscape.org from the site which is provided here. And there are 3 formats. One is a simple interaction format. It can be protein-protein, it can be protein DNA, or it can be your mRNA, mRNA. So whatever you represent as a protein-protein, protein DNA, the mRNA, that you can represent, and you can put it, and just node 1, node 2, node C, node A. In that way, you can represent in a simple interaction format. You can represent the nodes, whether it's a protein, whether it's a gene, whether it's a phosphorylated state. You can represent the node, and then you can give the age as a number, how much confident you are that those two molecule interact. It can be protein, it can be mRNA, mRNA. So whenever it's a mRNA, mRNA, it will be a negative association, or whenever there is a protein DNA, it is a positive association. It can be positive, it can be negative as well. And if it is a protein-protein, which is a signaling proteins, it's always a positive interactions. So you can represent the positive interactions, which can be protein-protein, protein DNA, it can be mRNA, mRNA. So all those interactions, and you can give some strength, which is a confidence value, how much confident you are that they are interacting. So this is how the cytoscape looks like once you download it in your local computer, which is very easy to use. And the good thing is that it's like a brushing tool. You can represent all your interactions, all your nodes, the different species I was talking about. It can be protein, it can be mRNA, it can be miRNA, it can be metabolome. You can represent it as a different shapes of a node, and you can use the age attributes, different age attributes to represent the different interaction. Suppose, for example, the APC, you use APC and you see all the interactors coming with APC. There are different ways you can represent. This is organic layout I'm showing here. So finally, within a cytoscape, which is very important tool to study the interactome, and there is a network analysis tool is there, which allows you to identify all the network properties which you have generated. So for example, the clustering coefficient I was talking about, you can generate clustering coefficient. So it means that the interactors, they interact upon each other. So that's why it's clustering coefficient of 0.463. Then we talk about a shortest pathway, then characteristics path length, the average neighbors. So in this way, all the graph properties which we need to study or which we need to report in our day-to-day basis, you can get it from the cytoscape. And within a cytoscape, there is a network analysis tool. And within the network analysis tool, you can identify all the properties of a graph. And interestingly, there is a node degree distribution is there. So once you click it in your node degree distribution, it allows you to give the whether it's a scale-free network or whether it's a synthetic data you have generated. So this particular tool allows you to study the network as you have generated in your study. So at the end, in summary, I talked about the overview of single layer omics. I talked about a little bit about genomics, proteomics, mainly the phosphoproteomics, the interaction proteomics, metabolomics, and how these genomics, proteomics, and metabolomics, how you integrate them to form a multiomics layers, and where you can study not only the interspecies interaction but also the interspecies interactions. I little bit touch about the graph theory, mainly the degree, mainly the half proteins, mainly the scale-free network, and at the end I little bit touch about the cytoscape for network visualization and the network analysis. So these are my students, and we do go for a trip, and yeah, we got funded from our DBT, ICMR. Thank you.